Fermentation is known as the chemical breakdown of a substance by bacteria, yeast, or other microorganisms. Yeast is a common substance that is known to undergo a fermentation process to produce ethanol. This process was one of the numerous ideas on our STEM team members' minds at the beginning of the year when we all met for the first time in August 2016. From there, team members conjured up experiment ideas that included mushrooms, bacteria, hydroponics, and tardigrades.
Some challenges that I faced in STEM, along with the entire team, is just coming up with an experiment. So at the beginning of the year, our mentor came to us and said that our focus was going to be on tardigrades. And tardigrades are really cool, and we love them, and we love the idea of them and the fact that they've been to space before, but it was really challenging coming up with an experiment to incorporate them within. So everyone had different ideas, and all of them were great ideas, so it was really difficult to narrow them down and pick just one experiment.
Tardigrades, also referred to as water bears or moss piglets, are microscopic, extraphilic invertebrates that can live for as many as 120 years. They're about 0.5 millimeters to 1.2 millimeters in length and have 8 legs, with 4 to 8 claws on each leg. Our team became extremely intrigued with their ability to survive in extreme conditions.
The super cool thing about them is that they can survive basically anything from extreme temperatures, low and high, to extreme pressure, and they've even survived in the vacuum of space, which is really cool. Tardigrades were great for our ISS experiment because tardigrades are basically indestructible, which means that they will be able to survive the rocket launch going up with all the shaking, and they have survived in microgravity before, so hopefully they'll survive again.
Interestingly enough, cohabitation with ethanol is one of the few conditions that is toxic to tardigrades. We began to ask ourselves, what would happen if we sent tardigrades together with yeast to space? Furthermore, what would happen to the yeast production of ethanol through fermentation if we combined tardigrades with yeast in a microlab? We hypothesized that if yeast and tardigrades are propagated in conjunction under microgravity in a glucose solution, then the tardigrades will alter the ethanol production rate of the yeast. We will test this by constructing a microlab, a small payload about the size of a soda can. But first, CAD diagrams must be created.
A CAD diagram is a program that we create the microlab. We create this microlab in the computer, a 3D model, and then we send it to a company to be printed off. So on the computer, we used a program called AutoCAD Inventor, and basically we made the microlab from scratch and using that program to design the microlab.
Once the 3D printed microlab is received, the microlab is equipped with 2 fermentation chambers lined with silicone sealant. The chambers are separated from the electrical work by a polycarbonate window. The polycarbonate window is made from a transparent polycarbonate material, which we have crafted to fit on the open side of the fermentation chamber. We have carved out 4 holes into the window of our 2 ethanol sensors and our 2 temperature sensors. Gore-Tex sheets are glued on top of them, functioning to protect them from the glucose water solution that will be distributed by the 2 water bags.
So if we didn't have water bags, then the water would not have anywhere to go into the actual fermentation chamber. So to construct the water bags, first we cut out little sheets of clear plastic, and then we take a press and we actually use heat to press the pieces of plastic together to create the water bag. And then we take an X-Acto knife and make a hole into the water bag where we then feed in a tube so the water can be put into them.
The water bags will be constructed from vinyl sheeting and tubing, then assembled using Aqueon silicone and corium sealant. The seal is verified through a water leak test and further tested by covering the hole and squeezing the bag relatively hard and shaking the bag. The edges are sealed using a PFS 200 impulse sealer, and the corners of the bags are then covered with a silicone aquarium sealant. We also sewed a Gore-Tex pouch to place both water bags in for extra protection.
So the main problems that we faced, one big one was that multiple times we would try to press the bag, but a lot of the times holes would open up on the edges. So a lot of the times we would have faulty water bags. We probably made, I don't know, at least 40 water bags over the course of the experiment.
There are numerous levels of containment in our microlab. The first level of containment is the aquarium sealant that has been completely coated on the inner walls of the microlab. Next, the fermentation chamber is sealed with the custom-made and glued-in polycarbonate window. Thirdly, our whole and complete microlab is sealed as we screw the system together with 8 strategically placed screws. Before we run this experiment on the International Space Station, a control experiment must be set up and ground data must be gathered to ensure that the experiment will work and provide accurate results.
So the purpose of our control experiment is to replicate the one that we are sending to space here on Earth, so that when we get results back from space, we can compare it to the Earth data. So the Earth data will act as like a null for the ones that are in space. So the setup of our control experiment is the same exact microlab that we use for the flight unit. So we printed an exact replica and we did the exact same thing as we did for the flight unit, but instead we put it inside an incubator set at the same temperature that it would be on the ISS, and we're running it in there. So it's the exact same thing as what's in space, except with gravity.
Obviously, there are challenges in creating this control experiment due to both user error and external influences. For example, after running the group experiment for 16 hours, 0.5 milliliters of glucose water solution from the bag was used. 18 milliliters was originally inserted into the water bags, so only 7.5 was left. This had the control experiment team debating if more solution needed to be inserted before the experiment was sent to NASA. However, since only 15 milliliters could have been pumped in the first place, there were still 2.5 extra milliliters in the water bags. Many of such struggles, such as starting over, redoing entire experiments, pausing and resuming, and correcting miscalculations, are prevalent not only in the control experiment portion of the project, but in all portions of the project.
What challenges have you faced?
So in the process of making the microlab, we've had 3 to 4 different versions of the microlab, and that's due to different errors that we've had, or just making it simpler for us to assemble and put together the whole microlab as a whole.
Nevertheless, in the words of STEM team member Abby Irby, failure is not an option, and our science team commits to trying again. The difficulties and obstacles that come with creating an experiment are offset by the rewarding data and conclusions by the end of the experiment. In addition, it is through this trial and error that we have created an experiment that will test the effects of the presence of tardigrades and microgravity on the ethanol production of yeast. One chamber will contain only 0.1 grams of yeast, while the other will contain both 0.1 grams of yeast and 200 live tardigrades. Each chamber will also include a temperature sensor and an alcohol sensor. The chamber containing yeast alone will function as a control experiment. Its growth, ethanol production, and temperature will be a means of comparison for the behavior of the chamber containing tardigrades together with yeast. In order to properly culture each sample of yeast, 2 RPQ1 water pumps will draw a 0.29 molar glucose solution from 2 water bags and distribute it to each chamber. The solution remains a 0.29 molar glucose solution with 17.5 milliliters of water and 2,808 milligrams of glucose. A camera will also photograph the fermentation chambers regularly. The temperature and alcohol sensors will provide quantitative data that is representative of the ethanol production rates of the yeast in each fermentation chamber. The pictures taken by the microlab camera will provide qualitative data that will support the pre-existing quantitative data pertaining to the yeast ethanol production rates. Software and electrical are responsible for the functioning of this PCB board. They have worked together to ensure that the parts of the experiment, like the sensors and the camera, will function remotely the way that they should.
The software actually tells, the mechanical builds the experiment, and the software tells the experiment what to do at what time. So basically we tell the parts what to actually do.
Yeah, I know. If I put it back, it would like turn blue. Exactly.
It's going to change.
Yeah, so it is what it is. So what, that's just commented, right? So why are you?
Yeah, I mean, that's what Jared does. Well, as you can see, it's repeating itself, and it's like not gonna work at all, 100%. So we commented everything, and then from Mr. Nadir sent us code with like a replace, and then so we copy and paste that, and then we are taking back this set of what he wrote, the part, and then we are also adding replay for sensors and pumps, although we're not like doing pumps like multiple.
What problems have you encountered?
A lot. Mostly syntax errors and things just changing constantly. So in code, you have to write it specifically in a very certain way, or else it just doesn't work. Like none of it works at all. So we had to spend a lot of time trying to make sure that's right. And the experiment changes constantly. So every time it changed, we'd have to fix a little bit of it. And we keep going towards that, And as soon as we thought we were finished, I know Zach and I talked a lot, we're like, we're finished. And then they'd say, actually, we're gonna change it. So, and that went on for pretty much the whole year.
Like in the kitchen.
The cat will move in.
That's genuinely okay.
Can you check the So the electrical portion of the experiment has to deal with all the electrical components that we use. So we have different sensors and different pumps, and we wire it all together to a PCB board and make sure that all the power requirements are met, things like that. Power voltage, it won't be a full test unless we run it on a McMech and actually get the measurements on the screen. So we can start testing it now using the voltage supply and try to fix any errors we have, but ultimately we're going to end up testing it. So right now we're going to use the DC. So we encountered a lot of problems this year. Most of it had to do with our alcohol sensors. So we would have to stay after class and come in before school and things like that to test each connection point and check the voltages and see if they were acting as we thought we should see them act. And we had Skype calls to people. We would call Mr. Nadir in California to see if he could help us because he knows a lot about electronics. And just working with Miss Arvin because she knows a lot about how electronics works.
You don't know how long it was on sale?
Yeah. 3:30 Ashley.
What do you want?
Can we just like
We got this one. Oh, now when I tape it down and tell people, do not touch.
Well, yeah, I have a neon radio card.
In a normal class period, we do a lot of soldering, which is we melt solder, which is like a metal string, and then we connect different wires to the PCB board.
It keeps not wanting to melt, David.
We do a lot of documentation. We make a lot of electrical schematics, and then we troubleshoot. That's the main thing. Once we solder everything, we test it to see if it works, and when it doesn't work, we gotta find where we went wrong. Although
I thought last version that we saw, we had like
I don't know if that would be
before and
affected by
after
whatever we
photo
just
and take photo return.
it would have
Sensor, alcohol sensor.
Each part of STEM clearly contributes a fair share to the success of the experiment.
Because then it has to rerun.
That is awesome.
So I guess it's actually weird. I would get.
You didn't do any cookies?
Tuesday or Wednesday, just in case. What you can? What day can you get? None.
So we're reprinting this, right?
Yeah.
So we don't have to fill it with yeast and water now, right?
No.
We don't have to. So all we're doing is just removing the components.
So we are reprinting this part, so we can't even fill it today anyway.
Hey, you want me to take off the back?
Yes, just
You know, what would I do without you?
for these things. Let's bang them together.
She has that on video.
Remember that from Brian. You look great.
So we know what we're doing.
Yeah,
we like to think we know what we're doing. Yeah. Are you sure? Just don't ask us to make
Guys,
another water bag.
we're NASA, sir.
Make another water bag.
Please don't. Alright, Evan, make another water bag.
Are you gonna make another water bag?
No, you have to. You can't because you have to then take it out of the cup, which is keeping it in the dark.
Yeah, we're adding different things. And then
We're keeping our test tube. You have to keep it on.
why?
Because it's how you're measuring the temperature every single time. If you don't keep that on there,
Just not keep that on. That mean we don't.
no, because we're keeping it in the dark. That's how you're keeping it in the holder. If you have to constantly pull it up and out, then it's not gonna hold anymore. You're gonna ruin the cup, and then like we have no way of holding the test tubes up anymore.
That is so hard. Yeah, we're trying to that thing.
So we got it on there the last 4 times. It's not that bad. It's become easier after you do it 1 or 2 times.
Just use that in the real.
Once the experiment runs, several useful real-life applications can be drawn from the experimental conclusions. Among these are, the bacteria has already been tested in microgravity, with findings that certain species grow twice as fast in that environment. Yeast is a fungus, not a bacterium. Therefore, this experiment gives us the opportunity to retrieve newer data pertaining to fungi's growth rates in microgravity, and to see if they will mimic those of bacteria. The data retrieved could also potentially be useful in better understanding the future of agriculture in space. This field is continually gaining relevance due to microgravity's apparent aid in plant growth. Just like fungi and bacteria, plant growth in microgravity was found to be accelerated compared to Earth. Our data could help scientists resolve the question of why plants grow faster in microgravity. If yeast produces in microgravity at a faster rate than on Earth, there will be an increased level of carbon dioxide production, and fungi assist in the production of nutrient-rich soil as they work to decompose organic material into usable nutrients. This is useful in plant growth as well. Thus, our experiment could result in the creation of fertile soil and an overall habitable environment for plants grown in microgravity. Lastly, fermentation of plant materials to make ethanol has been greatly promoted as a means of producing sustainable biofuel. Through our experiment, it is very possible to discover strains of yeast that are able to increase and decrease in the amount of ethanol produced.
What people have helped us in this experiment and how?
One example of that is a man named Mr. Grunach. He works for Honeywell, and I actually work with him, and our mechanical engineers work with him to print off our CAD designs, our actually 3D printed, customized microlab.
Had them all running
In the beginning of
the 2 A's, the outputs.
Show me what that would look like.
Yeah, 2 A's. It would be connected
feels so
here,
crazy.
and then one, and then
I can't, like, think about where you're surrounded.
I can really picture. I'll draw it.
Alright.
Maybe I just had them all
Drawing.
resistor grow.
It's like if, then,
Those pen
no.
the holes.
I apologize my handwriting. Just like let them check that, you know, like is it multiple to it? Yeah. Is it let them ask it, and then if yes, then you go to here, do the replay, and then probably like let them come back here later, and then like if not, yes, and then if not, you just like wait. Yep.
Alright, that makes sense.
Glad.
I'm assuming
You like solder?